Thank you for the invitation. It's always a it's always a pleasure to share the research with everybody, children, scientists, or general public, or in this case, lots of naturalists. And talking about Santa Barbara in Southern California, actually, the whales I'm going to talk about, most of them, the one that we know pretty well, are actually migrating to Southern California. So you might see the same whale we study if you go out. Okay, so the idea was to talk about blue whale and their importance in the ecosystem and how we could even get to use their presence and their feces to look at climate variability. I'm very excited to share with you some new data we have not published yet, but before that, I need to talk about the population. Now, where are those? What are those blue whale? So this is the best diagram or figure, I think, to explain a bit about blue whale movement. This from Bailey et al. In 2009 from Bruce May group in Oregon State University. And so these are tracks from satellite tag that were um, that were put in different area like Cape. You can see on the top Cape Mendocino, Farallon, and Santa Barbara, for example. So if you look at the orange colors, these are movement of a whale that was tagged in Santa Barbara, and you can see many of them are going to the Costa Rica Dome, which we know it's another winter ground for those blue whale, those Northeast Pacific blue whale, but also in the Gulf. And and so I was telling you that many of our well-known whales, we know they by comparing picture with John Calabukidis. So we know many of them like the Southern California bite and also Santa Barbara and Channel Island area. But others are going, many others are going to some place we don't know actually. So we think it could be that they stay off uh, the Baja Peninsula all year round. We know there's blue whales are all year round there, and or up north, maybe around the Aleutian Island where there's very few people, you know, doing cetacean survey because of the weather and because of the cost also. So that's basically, the whale we study in the Gulf California are whales that are from the Northeast Pacific and they migrate during the winter, spring in the Gulf and also in Costa Rica. And they also feed during the summer in California and other area north. Okay, so why the Gulf California is important? Well, it was known to be a nursing calving area since a long time, since Leatherwood actually the first time I think it was 1982, he went in the Gulf and saw mothers and calf. And but then we realized they also feed over there, not like the humpback whale or the gray whale, who rarely are seen feeding because where they are, there's not much food actually. But blue whales go somewhere else. They go in the Gulf and they are in the whole Gulf, but they choose some particular area where there's a lot of krill. And the Gulf California is known to be a very productive area compared to other upwelling areas such as California during the winter. During the summer, the biomass of krill go down, but during the winter, there's a lot of krills around. So they take advantage of that. And I think that population, because of that, because of their migration to two very productive areas, the Costa Rica Dome that I showed you in the Gulf California, means that they feed all year round. They need to feed all year round to for their energy budget. And then later we found out that they also reproduce there, but it was expect because a female will gestate during almost a year, about around 11 months. So wherever the baby is conceived, is will be close to where the baby is born, right? So it was kind of obvious, but we never saw a blue whale getting birth, a birth. I don't think we've never seen that. Just to give you an idea, for gray whales, it's been seen, especially in the area inside the lagoon where the mother and babies are. That's where they calves. There's maybe around 20,000 gray whales that gathers in these 3, 4 lagoon in Baja California. And in contrast, there's about only 2,000 blue whale, 2,500 blue whales. So the probability of seeing a calf is already low, and probability of seeing a bird of a blue whale, I think it's zero. But hopefully, someone one day will be lucky enough to see that. So Gulf California during the winter spring is then a very important area for their uh, biological cycle, right? They are born. Some of them are born there. They come back and also give birth, reproduce and then give birth. They feed. They know the area very well, and they're very highly fidel to area. That's why we can follow them through time for such a long time. Show you some example. How do we do that? Well, we take pictures, right? And uh, the way we identified blue whales in the past, the first publication was basically based on the pigmentation, coloration of the animal. But I found, when you get to 700 blue whale, it gets like really complicated to just look at pigmentation. So we decided to categorize the catalog into dorsal fin because they have a very distinct type of dorsal fin. Some of them are very hook, other are mark, some other are even chopped totally. Others are more like triangles. So it's easy to get to the part of the catalog where you want to compare the whales you just photographed, right? And then of each of these type of dorsal fin, we have 6 types. We also categorize them each into pigmentation from almost white to almost black and in between, right? And so why do we have to take picture of the body and including the dorsal fin? Is because not all the whales show their tail. Like we calculate around 25% like to show their tail. And we don't know why. Some females, some males. It's something we never figure out yet. Why some show their fluke and other they don't, but that's the way it is. And so just taking the picture of the fluke, which is much easier to identify, but we wouldn't be able to identify all the whale. So this new classification help us a lot in the field. We have our own little catalog of the most uh, fidel animals, and so we can know right away. I mean, very quickly who are we studying, who are we following for. And so, but when you see a whale at sea like that, unless it's accompanied by a small calf, a small individual, we have no idea if it's a female or a male. So, during many years, especially at the beginning of my career here as a professor and researcher, so we took biopsy, which is um, which is taken with a little narrow that is propulsed by a crossbow and a special type of metal that has some teeth to grab the piece of skin and the piece of blubber. It's very small sample. It doesn't really bother the whale. It's more the approach, to get close enough to shoot and hit the whale at the right place, let's say. But after a while, when we got enough information from most of the whale, I stopped taking biopsy because I don't really like to personally. It's not in my method to do these kind of approach. But we did that several years, at least 15 years, to get a good catalog with the sex of the animal. And with this biopsy, with this piece of skin and blubber, you can do many, many, many different study as well. So, but in general, if we think about the sighting history, we almost got 30 years of data, a little bit more than 30 years. I always have to count back. One, two, yeah. So here we see through the years the different individual each line, and in blue are the males, and in pink are the females. And within the female, you'll see those square, black square, which indicate that that particular female, that particular year was seen with her calf. So we can look at the calving interval. You can look at uh, so many different things in terms of their biology that we still don't know very well. For example, calving interval is estimated to be around 3 years. No, every 3 years a female whale will give a birth to a calf, but it's not the case for all the whales. If you look at the data right there, some of them are also giving calf every 2 years for a while, right? And others are seen, they're in pink, they're seen many, many, many years, but they come with a calf every 5 years or 6 years. So it really depends on the individual, and I'm still have not figured out. Uh, I'm getting close to it because we are measuring the animal now, and I thought that the whales that were very productive that would give a calf every 2 years, for example, like the number 250, was a young whale, not a young healthy whale. But I'm finding that these female that bring to life calves every 2 years or every 3 years, they're usually very large, so probably old and experienced whales. I think it's the case in other mammal too. And there's other little square with with a little circle in it. That means that this whale in particular was first seen as a calf. So, for example, this one here, number 67, was born in 1994, sorry. And now he is, I can see 30 something years. So we know some of the individual, we know the exact age when it was first seen as a calf. And the same with the female. And for example, last the one that are 22 years old, you can see as they have had their own calf as well. But there's very few female after 30 years that were born that we photograph when they were a calf that has came back with their own calf. So it's not we don't think they all died or something. We just think they might have in Costa Rica dome, for example, which was shown by looking at the isotope in the Baleine place by ex-student Geraldine Bousquet. So we know that some female do come in the Gulf and then decide to go to Costa Rica, for example. But still, after 30 years, we do have very little information on the minimum age of the age when females start to reproduce. Okay, so these are more general kind of data based on the knowledge of the individual through time. And then, as I was explaining, like for example, this female 250 had three calves in 2 years interval, and one of them came back and also gave her first calf. And this interval, the smallest we have so far, is 9 years. And the estimate from the whaling period is between 5 and 10 years. So it kind of fit into that interval. So, so we have over 800 individual. Uh, about half of them are seen regularly. The other half have been seen only once. So we're not really tracking 800 different individuals. I mean, we try, but some of them were seen only once, and we've never seen them again. So the focus is more on about the 400 that are more fidel to the area. And so about half of them, the one that we know the best are sex. So we know if it's a female or male. And we also have several calves. But as you can see, it's a long-term study and it's very slow. So, when people describe me as an expert on blue whale, I usually say, well, no, no, I'm more like a very patient and passion researcher on blue whale because it's it's, um, it's very slow to understand, even these simple basic biological data. And I think they're very important, and that's why I'm doing this with all the students. That's why I'm continuing, but I'm getting old, and whales lives a long time. Anyway, so also, okay, so we know the individual, we know if it's a male or female or a calf. Some of them we know their exact age, and we've just published about 2 years ago a new method that we worked for many years to measure the whales from a boat using the photo ID pictures, but using a laser range at the same time. So we know the distance, we know the camera, this focal, etc. And then we can measure with scale the length between the blowhole and the dorsal fin, and then we can extrapolate to the whole body with model that we also work on from aerial pictures. Anyway, so this is the distribution of the 130 whales, 160, I think, that we measure. You can see in black the males and gray are the female, and you can see right away that the females are much larger. The maximum size with the maximum number is much higher than a male. And the largest whale that we estimate was about 28 meter, and the smaller one was 7 meter, which was a calf. And it's kind of it quite fit pretty well with the estimate of the calf size when they're born in the North Pacific. It's about 6 to 7 meter. And then we did with my good friend Tim Girodet. We use a Bayesian model, and you can see here sometimes we can measure or take what we call photo sequence of an individual several times during a season. So when we are lucky enough and have three different measurement, the error really decreased quite well. And so the error for three photo ID photo sequence by season is about 1 meter. So for a whale of over 20 meter, 1 meter is not that bad. Anyway, so that's what I'm I was telling you about the female, the older female or the larger female seems to be the one that produce more often calves. It's because of this data that we can get to that. Oh, I want to show you here. I don't know if yeah. So that's the way we do. We drive alongside the boat, the whale, and then shoulder to shoulder, someone is taking the whole sequence of picture and the other one is taking the distance. And we need three distance to be more precise. Anyway, so that's the okay. Now, if we were just doing that, it would be hard to to publish and get funding because it's such a slow, long-term study. So what we did was at the same time like we take picture, we know the individual, we try to well, not we try, we started to take different biological sample. The first was biopsy. It was the basic thing for genetics, right? But then I realized oh, I didn't write it well. The slough skin, piece of long piece of skin that floats at surface. And I learned about that by studying sperm whale actually, and I was really interested because we can also use the skin to identify the sex. No, not the sex. Sorry, the haplotype. So it's very useful, and there's absolutely no. I mean, you're not interfering with the whale at all. You're just following the whale, and you look for the skin in the floating in the footprint. No, the footprint, the big circle that forms when the whale goes down. And then I start to see feces because in the Gulf they feed a lot, and we see a lot of feces. And I thought, why didn't I start to take sample way before? But it was progressive, you have new ideas, and so with the feces we can do a lot of the study type of analysis too. And then the blow, which is something we started. Actually, we were the first one to publish, sampling the blood to look at the microbiome or bacterium, etc. So all this to say, okay, in terms of the long-term study, you can see how, the digital photo ID started in 2006. So that's when we start the measuring. That's when we start the photo sequence because it's much easier to work with digital. I mean, it's necessary actually. Same with the body condition index. I'll talk about that a little later. And you can see also the drone how far in the period it was available for us. So, but still, now we have almost 10 years of data. But compared to the other kind of biological data, it's quite recent. And so, but because of feces has such a incredible potential, I want to focus on some of the research we did on feces because in 1 hour I cannot cover the whole study of 30 years, right? Anyway, oh yeah, and many big people ask me about what kind of technology really helped to improve the research on whales. So I would say, like I just mentioned, is the digital. Can you imagine in the past we were taking the pictures in black and white because it's cheaper and you can do your own enlargement and what part of the whale you want and all that. And so after the season we didn't know if we had a good photographic season because we wait until the end to do the dark room, which is a lot of work. So that's an example of the same whale, in 96, black and white, 2005, digital. So that changed our life totally. It changed our life because we can look at the picture with the camera in the field with the catalog, to be sure and look at the different pigmentation. that we need to positively do a match. And so it's so incredibly useful to use digital. I know, I imagine many of you don't even remember the black and white. I don't know, but I do because I worked a lot in the darkroom. Anyway, so that's one of the most important to me. And the other one is the drone, the accessibility of drone. But this picture, this thing there, it's not a drone, it's an helicopter that was maneuvered by someone, of course, but that used gasoline and had really, it was really, really dangerous. And so my friend Karina, she's the one who, because we were talking about the blow smell bad, and she said there must be bacteria and we should sample it and blah, blah. We discussed. And she came up with this idea way before the drone. And she said, "Oh, I'm gonna find the money, I'm gonna buy it, and I'm gonna drive it. It's easy." They told me it's easy, but it turned out it was not easy at all. And so we had to hire a friend, someone that was an expert on that, and to join us on several day trip. Imagine, to sample the blow. But anyway, we were still the first one who started this type of analysis from the blow of the whale, and that gave us also this. Maybe you never heard about that. The ignoble. It's the improbable kind of study that that has been done, and it was basically because they were calling it the whale snot, no, which is not well bred actually. But anyway, and it was really sexy because we were using that helicopter, that terrible helicopter. Anyway, okay, but then when the drone were available, we really start seriously to sample the blow, and also meanwhile, the genetics got so much easier to look at the microbiome or the bacteriome, and with this mass sequencing and all this, which happened more or less at the same time. So yeah, Carlos Dominguez Sanchez, he did his PhD, and he um, he sampled over 20 whales different times during the season. And there's an article that it's in revision right now. So he looked at some bacteria family, and he analyzed a core of, what was the family bacteria family the most common that blue whales have. But there were some of them that differs and had different bacteria, actually dangerous bacteria that was very different than the rest of the individual. Anyway, so we're now working on what is called fungoma, which is the other part of the respiratory pathogen, which is the fungus. And yeah, we're finding very interesting stuff. Anyway, so that's and with the drone, many researcher also like showing right here, but they use drone to measure the whales or to measure their body index, using aerial pictures of the whale when it's totally straight at surface. And Hasivi, which is part of that 3D movie film, um, she developed an index that is based on photogrammetry, and very easy to not easy but much more precise, because it finally gives us a number. Is it over or lower than zero? So it's a good condition, or low, or bad condition, or normal condition. Because I'll show you the other index that we use is more subjective, based on visual pictures. No, in that case, it's a measurement and it's very precise. Oops. Um, okay, so whales are considered sentinel of the ocean health. So I'm going to use the some of the analysis we did with the feces to show you why, for example, blue whale could be, or is actually sentinel of the Gulf. Oh, well, the ocean, but in that case, we're in the Gulf. So the feces of blue whales are very easy to sample, blue whale and fin whales, and also humpback whale. But the whales that feed more on fish, like dolphins and or squid, like sperm whale, their feces is like liquid. It's very difficult to sample. You sample water basically. But in the case of blue whale, you do sample chunk, really big chunk of feces. And you can, I mean, so yeah, depending on how you preserve them, you can look at the prey. And a student in 2010, she compared fin whales and blue whales, and she looked at the gene that associate with not the gene, but associate with krill or fish, teleos fish. And she found that, yeah, fin whales feed a lot more well, the feces had much more DNA of myctophid than krill or afenaf. And blue whales, out of 40 sample, 39 also showed myctophid, which is the lanternfish. Very, very, very abundant, that little fish that move in the water column day and night as the krill. And actually, myctophid larvae feed on krill, so it's quite interesting. But anyway, we were very surprised that blue whale also ingest, some part of the mycmus, some myctophid. We don't know if they're larvae or adult, but all the sample except one. I mean, it's quite common. That's the idea. And the other interesting thing is to look at the parasites. In the feces, you find adult parasites sometimes. It's not quite common, but you do find them. So some student got to the genetics and look at the species, but you also find eggs of helminths, element in general. And then you can use the number of eggs per gram of dry feces and do comparison between individual or within time and period, etc. No, very interesting study. And so one of the students continued that topic, and she used a different way of sedimentation of the eggs of almond eggs, and she found very interesting protozoa, actually human protozoa, like amibs, entomeba, ballantinium, and giardia. And these protozoa should not be in the whale feces. It's human. And so that is indicating that in part of their migration, in part of their habitat, they are contaminated by human feces. So that, to me, is quite interesting, important to maybe in the future, if not, other students continue in that subject, an an over number of one of those could mean something about the immune system. Okay, so again with the almond egg, so here we go. Here is all the different sample of different individual. And you can see some of them are very highly parasites, very highly concentrate in almond eggs. And we try to see if it has something to do with the female lactating or male, between male and female, juvenile, la la. And no, we didn't find any clear tendency here. So some of the individual are very highly parasites compared to other. And she also compared with fin whales. And blue whales had 100% in all the sample we found almond egg. In fin whales, there was only 70%. So there's something interesting here to look at too. Anyway, so that's one of the research about what's what happening in their, well, as a proxy of health, obviously, because that cannot explain everything about a whale health, but it's part of it. I mean, in human, we do that. Actually, in Mexico, it's it's very common to bring your a little sample of your feces to see what's going on if you have amebs, antomeba, justamente, or other things, right? So it's a way to look at the health. And then another student, now that we have that mass sequencing, the next, what's called next generation sequencing, which is basically you look at the at everything that is every every part of the DNA or every organism that is in the feces. So that includes their prey, that includes their own cells, that includes phytoplankton and even spore of trees. And I mean, it's amazing what you find when you do the whole sequencing. But when you look at the possible prey, this student, Vladimir, he found basically euphausids. In all the sample, like euphausids is like 98% of the DNA. But in all of them, we don't see very well, but there's all of them have as a little blue line. So all of them have also the myctophids, and some of them, some sample were completely, almost completely contain DNA of myctophids. So that's interesting. Why some individual in some years, it's not common, obviously, we can see that, but it happens. So it gives maybe a little bit more information on how they get to feed on myctophid. Maybe, during the time they were feeding, the myctophid were really feeding on krill, or they were at the same level, or whatever. But it varies. We can see that. Okay, with feces, we can also look at the hormones, and that was done first by people from the East Coast on right whale. And so, but their method was to freeze the feces, which in Baja it's a little complicated because I mean it's possible. We've done that with biopsy, having liquid nitrogen, a little tank on the boat, but I thought it would be complicated for feces because anyway. And so while I was looking at the feces under a dissecting microscope, looking at the mandible of euphausids when I was a student, I remember my petri dish dried very fast. And then when I learned that steroid hormones are very stable and that some people, well, mammals from Africa, I mean, they were doing hormones, steroid hormones on feces from giraffe and rhino and elephant. And they use also dry samples. So I said, well, we just put the sample in a petri dish in the boat and it will dry. And so that was a very interesting new method, much easier for us. And so that's what the student did. He analyzed several individual and pregnant, I mean, resting individual, lactating. Well, no, first individual that we knew was with calves. There was one male. And then we had to differentiate between the resting female that are not lactating, but they might not be pregnant, and progesterone and corticosterone. I mean, in several whales, more than half. No, not more than half, or 25% actually, had very high concentration of progesterone, and that's what you expect with pregnancy. As soon as the ovule is with the spermatozoid, it doesn't take very long. The progesterone start to increase very fast. So it's a way to know a little bit more about our female, the one that are not with calves. Are they pregnant or are they resting? it's very, very interesting data. We are now in the lab here because it was complicated to send the sample to Alaska where we had the collaboration. But now we do everything in the lab. We have all the equipment. We're looking at testosterone also. Anyway, so with that, the pregnancy, the lactating female, uh, there is a characteristic of lactating female that is that you see them very skinny. You see their vertebra process quite clear. And actually, sometimes in the field, when I see an individual with a calf that doesn't look like the big mom I'm used to with the vertebra process and the sloughing skin, and I usually say, wait, this is probably not the female. And by waiting and following the individual, then we realize it's not. And all of a sudden, that big mama come, and it looks exactly like that, right? Wow, it's already 7. Okay, but aside that, in 2015, we start to note that many whales were skinny. And one thing is to say, well, I feel the whale are more skinny than having an index, no? And so there was an index of body condition that was already developed for gray whales and right whales. And we used that uh, to look at our individual. First, I want to mention that publication by I don't remember the name, but it's with John Kanabokidis data in California. They also note something similar uh, from 2015, actually, and they correlate it with the PDO, which is the Pacific Decadal Oscillation. And during those 3 years, the index was over zero, and they kind of explained that during that period, whales had probably less food, less biomass, and were more skinny. So they observed something very similar as we did. So which is interesting because we're going in the same direction. Although they use a different index, it doesn't really matter. We use 3 characteristics: the vertebra process, the postcranial depression, which is very rare, but we see it sometimes, and the lateral depression here. And so these are the general data. We use so it's a visual index with photo ID, right? So we can use data from 2006 is when we start to use the digital camera. And you can see there's a tendency, not just from 2015, there's a strong tendency of decreasing in the good body condition index compared to the regular or even compared to the bad. And so I was also seeing, okay, and then a good friend of mine that works with crews from Scripps and Cal Coffee Grid of Baja California. She compared she compared old data like 2097 to 2097 to 2014, and then for 2014 and 16, and she found incredible result of krill biomass decrease 95% during those 2 years, the last 2 years she studied, 2015 and 16. So it also showed that even close to the Gulf, during where they migrate, when they go up north, the biomass of krill also went down. So, but that was during El Nino, that was during the blob, 2015, 2016. but but what we found is like a tendency. So knowing that knowing that with the blubber thickness is kind of representative of the index of body condition. And during the whaling time of fin whales in the North Atlantic, They were capturing whale, measuring the blubber, and looking at the pregnancy. And so they found that as the blubber was more thick, the proportion of female that were pregnant was higher. No? So obviously, if a female has not enough blubber, will probably not conceive a new baby. It might be pregnant, but doesn't finish the gestation, or maybe not getting pregnant at all either, no? As this data is showing. So I looked. So what I did, this is not published yet. No? These are the good body condition index. How since 2006 it's going down, but it seems to come up again. And this is the reproductive rate, the number of calf photo ID per number of individual photo ID every season. And so you can see that is going down too, and not just in the last couple of years. I mean, it's really going down. And we applied the model here, and for the body index, at least we can see right now that it started to getting better in 2024. No? So, but the calving, I mean, the reproductive is not. It's just barely. I mean, it's down. So it's quite something that is concerning. Although the body condition seems to get better now, yeah, we have seen very few calves the last 5, 6 years, except 2018. No, in 2022 when we filmed, it was magic. There were four mother and calf all of a sudden, which is that tip here. But but generally the other years were pretty low. Anyway, so that is something that concerning us. And I need to finish with that. Okay, so whales, I can't read what's in there. Okay, yeah, whales also influence marine ecosystem by their feces. You probably have heard about the whale pump. And so we did a whale pump. Okay, let me explain to you. Most of the whales feed in the bottom below the ophotic zone, right? Where there's not much light. And the phytoplankton is up where the light is. So normally for the phytoplankton and krill and fish will use the nutriment, and then all of a sudden there's less nutriment. But the whales feed on krill or other stuff, and they feed in the area where it doesn't communicate with the photic zone. But the thing is that they defecate at surface. So that these feces actually bring a lot of nutriment to the photic zone, the zone where there's productivity, there's chlorophyll and phytoplankton. And so we did an experiment using bottles anchor in the bottom close to where we work, but close to shore, of course. And we use the water from the area with native phytoplankton. And we had a little bit of feces here, and a little bit more. So two treatment. And here you have the result. Without in the control, nothing happened. The control, we didn't add anything. So the phytoplankton was reproduction was doing chlorophyll, and after a while there was nothing. But after 4 days, the fifth day, we start to see an increment in chlorophyll and also in productivity, primary productivity. And look at this curve here with the most feces uh, uh, put in the experiment. The value is double. So it's incredible. I mean, the blue whale feces contain element, micronutrient with bacteria from their feces that it's ready to use by the phytoplankton. It doesn't have to go in the bottom and with the bacteria, all this cycle of the nitrogen. So it's very interesting. Some people have done that for Antarctica, for I think in the Bay of Fundy, and we are publishing. Well, we are about to publish in the Gulf. Now, for just to finish, we went further because feces are incredible. But so the question was, could well be indicator of climate variability? I mean, could the feces be useful for other things like variability in climate? And so because whales, blue whales are huge filterer, giant animal, we thought, well, and so to produce feces, they gulp and they take lots of water and they filter the water, of course. But we were wondering if the trace element in the water, trace and major elements in the water could be represented in the feces. So sampling of dust, because if you want to study, for example, the impact of the wind into the dust coming into the ocean, you need to sample the wind, the air, and it's very difficult. Imagine with nets and lots of contamination. So using the well is like it's just perfect. They're the mechanical way of sampling the ocean water. And so the idea was to look at the um, the aluminum and titanium and trace element that comes from the wind from the desert, because of course there's not many river in this area, so most of the trace element and major element are coming from the dust and the wind. And so in the sea, you have all this. You have phytoplankton. You have other organism transforming to feces. So if the feces produced by these whales could indicate this variability in terms of climate, and so we did the analysis of aluminum and titanium from feces over 24 years during a period where there was a high variability in the climate. In red is Nino, in blue is Nina, and the PDO, the Pacific oscillation, decadal oscillation is mixed in that. So it was enough large to have different high variability in terms of the climate. And so what the student find was in green is the aluminum, in blue is the titanium, and so the variation was very similar, and so we could conclude that there was from the same origin, the wind from the desert. They were not like contaminants from whatever, our boat or something, right? And then looking into more detail, taking the mean of aluminum and titanium and compare it to the one of the El Nino index, the one that is more in the central Pacific, you can see the high correlation. So I just want to finish with that whale species uh, are useful to look at climate. I mean, I wouldn't say climate change. We're not talking about CO2, and I want to get into that, of course not, but about the variability in the climate, because climate is always changing, right? In, in cycle, associated with the sun, including, no? Anyway, so I want to conclude now that the long-term, slow, boring kind of study are very useful to construct large time series that can be useful to develop new biological parameter to evaluate proxy into health of the ecosystem and also the whales, and also to evaluate the climate variability. So I think I'm going to stop here because it's been 1 hour, isn't it? So I had much more to say, but I need to stop.